Okay, hi everyone. I'm gonna make a start. Thank you so much for coming. I know there's a lot of people sick at the moment. Half of my lab is sick, so hopefully we all stay healthy. I'd love to acknowledge that we are on the lands of the Roundtable people and pay my respects to their past and present, and extend those respects to any Aboriginal people who might be here today. Also note that we are in Reconciliation Week, so we'll take a moment to reflect on the errors of the past and also look with hope to that where we can work together. So today we've got two talks. We've got a flash talk from Lindsay Tao, and then afterwards we'll have Associate Professor Luca Cocchi, who's all the way from Brisbane to speak today for the longer talk. So I'll start with Lindsay, if you want to come close. So just a very quick introduction. Lindsay is a PhD student in my lab. She is working in fMRI and looking at prediction models. And she will be giving a very short talk about one of her studies. Thank you, Lindsay.
Thank you. Sorry. Thank you. Hello, everyone, Lindsay, and thank you so much for the opportunity to talk about my study here. Really keen to hear everyone's feedback on this. And the talk is about understanding the neural mechanisms of prediction precision. And this work is supervised by Rob and Prof. And I am really interested in predictive processing theories. So this is a theory proposing that the brain does not just passively receive sensory input from the environment, but also actively generates predictions about its incoming sensory input. And a very important element in this theory is the precision of predictions, and there is precision of prediction error as well, but in this study we only focus on precision of predictions, and that's the inverse of variance or uncertainty. So predictions are not weighted equally, and predictions with higher precision or lower uncertainty are weighted more and will be able to exert greater influence on perception and learning. However, it is unclear how the brain understands the precision of its own predictions. And we thought that this might be explained by modulatory connectivity between brain regions. And we studied this using DCM, dynamic causal modeling. Dynamic causal modeling, very briefly, it estimates the effective connectivity between brain regions, and it's the directed influence of one brain region on another. So for precision modulation in visual predictions that we're interested in, we thought that it might be explained by some higher order cortical and or subcortical influence on V1. And for that, we focused on the insula for the cortical region and foveola for the subcortical region. And we collected 7 Tesla fMRI data. So this is just to show the 3 regions we focused on, the insula on the top and foveola in the middle, and then V1. So we designed our DCM model to test the connectivity between all of the different regions. And you might be wondering what kind of task we're using for this study. So we modified classic kind of grading orientation task. And so we showed participants Q at the start that predicts whether the subsequent grading orientations will have the same orientation or different orientations. To manipulate the precision levels, we designed 3 different Qs with different precision levels. So 80%, 60%, and 10%. We mainly focused on the 80% one and the 60% one, and the 10% one was included to balance the overall expectation. And so the behavioral results, we found a significant behavioral difference between the high and low precision conditions, and that's really nice. And the DCM results, so the arrows here show the modulatory connectivity elicited by high precision, and the different colors show whether it's excitatory or inhibitory. And so we can see this excitatory modulation from the insula to V1 and inhibitory modulation from insula to pulvinar and from pulvinar to V1. And we thought that we could propose a two-route feedback mechanism from insula and pulvinar to V1 that includes insula having a enhancing top-down predictions in V1 and insula inhibiting bottom-up V4 signaling in V1 via the pulvinar. And more results, so we're also thinking about whether we can use those connection strengths to predict people's behavior. And the answer is yes. So we could use the strength of these connections to predict the magnitude of the people's behavioral effect that's related to the precision conditions. So that's really cool. And in summary, we showed that precision modulation might be explained by effective connectivity between the brain regions. And more specifically, high precision elicits excitatory modulation from the insula to V1 and inhibitory modulation from the insula to V1 via the pulvinar. And we proposed the two-route mechanism for precision modulation, enhancing top-down predictions and suppressing bottom-up reward signaling. And we also showed that we could use some of the connections to predict behavior. So that's all for the talk. Thanks everyone.
Okay, thank you so much, Lindsay. Maybe you can take one question for Lindsay if there's any question. Natalia?
Can you talk a little bit more about why you chose insula as the top-down prediction model?
Oh yeah, good question. We had a lot of thoughts and discussions about which regions to choose. We chose insula because it has it's been shown to have a lot of salience and relevance related role in previous studies, and also there are some studies showing it may relate to the functioning of pulvinar as well, and it might have a top-down it might have top-down influence to V1, and we are really interested to see whether it has some kind of growing over studies. So we also really interested.
Just to add to Lindsay, that is why we chose the insula, but to clarify, with being top-down or bottom-up, that was decided by the model. The data that emerged from the data, so there's no constraints on the directionality. It's not priority. Hey, thank you, Lindsay.
Thank you.
Right. So please, when you get the message from Sarah asking for talks, please put your name in. We always want to see students give their talks. And also later on, we've got the Q&A session, so all students please stay as well after lunch. So, Associate Professor Luca Cocchi, he is based at the Queensland Institute of Medical Research up in Brisbane. I've known Luca for quite some years, maybe 10 years. We overlapped in Brisbane when we were both at the Queensland Brain Institute, so it's really a pleasure to have you down. So a little bit about Luca's background. He obtained his PhD in neuroscience at the University of Lausanne and Geneva in Switzerland in 2007. He did postdocs in amazing institutes, including here at the University of Melbourne, at the Melbourne Neuropsychiatry Centre, which I think now has a different name, has changed its name, and also the QBI. And then he started his own laboratory at QIMR in 2018, where he is the head of the Clinical Brain Networks group, and also the scientific director of the Queensland Neurostimulation Centre. He serves as an editor, or used to serve as an editor for the medical. He just stepped down from Neuroimage Clinical as a editor after serving for 6 years. So thank you for serving the community. He is recognized for his work in brain imaging in health and disease, and has applied imaging analyses in very clever ways, and I'm sure you'll hear about it today, to identify functional and anatomical brain connectivity underpinning different conditions, including schizophrenia, ADHD, and OCD. So I will stop here. I'm really looking forward to your talk. Thank you so much.
Thank you all. It's great to have you. This invitation to present some of the work that the lab is doing, and so I hope. So when Marta invited me, because I'm not a psychologist, so I was a bit tense about what to present, and so I put together a talk that is a mix between, what is more basic science and what is more clinical science. I hope you enjoy it. And so the talk, the title of the talk is a little bit ambitious. It's about what makes us human and what makes us sick. And I hope that I will give justice to the talk, and at the end of the talk you think, oh, I kind of get his point. So the first thing that I want to say, well, humans are amazing animals. So I put a picture here of this helicopter that we were able to, as a human race, to fly on Mars, and pretty stellar achievement. We are also able to create some codes that generate some beautiful sensory outcome, can elicit emotional and arousal change. And of course, science. We are compared to other animals. We are able to actually discover our own nature and the nature of the universe, and so on and so forth. So, and remarkably, I think one of the key organs, or arguably the key organ that allow us to do that, is the brain, the human brain. So all this is achievable, and what differentiates us from other animals is really has to be linked to something that is associated to this organ. So, and this I put just a citation from Kolk and Goldman-Rakic about the importance, especially the human cortex, to achieve all these things. Now, with great power come also great responsibility, and one of the issues that we also have is that there is disease that and these are particularly mental disorder, complex mental disorder, seems to be peculiar, or they have some characteristics that are only found in humans. There is some, I will show to you, there is some substrate of the human brain that is, cross species that is linked to this disease, but then there is also things that are human specific. And when you think a disorder like schizophrenia, when you think a disorder like anorexia nervosa or like that, there is something about the symptoms and the characterization of this disorder that is somehow human. And probably you as a psychologist may appreciate that more than other people. From an evolutionary perspective, just to give a sense, humans are up here. So there's this branch here on the evolution. Our closest relative on another branch, though, are the chimpanzee and the bonobo. So we are kind of higher up in the evolutionary, hierarchy. And the important thing, I will show some results about chimpanzee that are really our closer relative in terms of evolution. About the brain, so again, other facts. So the human brain is around 1.5 kg, 86 billions of neurons. And again, just to give context, when we studied, there is a lot of studies that are done in animals to understand. But if we look at the chimpanzee, is 4 times smaller the brain. You can see that there is gerifications, but significantly less number of neurons. And then when you go down, there is a smaller and smaller. And when you look at the mouse, that is an important animal model in medical research. Well, that's almost 4,000 times smaller than the human brain, and has around 70 million neurons. So it's a big difference just in power capacity between these two these two organs. Also, this is a quite famous paper from David Van Hessen showing the evolutionary expansion of the cortical area of the brain. And particularly what seems to be the case is that there is a greater expansion in the, what we call the TPGA area here at the back of the brain, and then also in this anterior frontal region. And these are the region that evolutionary speaking are the more expanded in humans. Another interesting observation is that is looking at the myelin maps of the brain. So myelin is basically this lipid membrane that is a proxy of speed of communication in the neurons. And so generally the sensory motor area are very highly myelinated. So, and the associative area, so linked to this complex cognitive process, have generally a lower quantity of myelin. So you can see that in human we have, this is the motor cortex, is a region highly myelinated. But then when you go down in the evolutionary tree, you can see that the level of myelin is becoming greater and greater. So there is less and less region, if you do some inference, that are linked to really cognitive thing, and more region dedicated to sensory motor, so sensory intake and motor outputs, to the point that is the mouse brain, the majority of the mouse brain, if you just take this proxy, is basically dedicated to sensory motor response. And this is another slide from a dear friend, Martin van der Heuvel in the Netherlands. What he did, he basically looked at so this is structural connectivity that is represented here. So is the anatomical wiring of the brain. And he basically looked what are the connections that are human specific and the one that are present in our closest relative. And here it looks in the chimpanzee. And what he found is that, the red are the human-specific connection, and then you go down, there is the enhanced human development. So these are connections that are also present to some degree in the chimpanzee, but are somehow significantly stronger in humans. And then there is a shared connection. And again, what he was able to show is that again, there is area connecting or connection connecting the frontal areas. There is the language cortex that is also very specific to human, how it's connected. And again, this medial wall region that are particularly connected in a different way in humans compared to the chimpanzees. And here I want you to remember, just for later in the talk, this particular area here. So it's called the orbitofrontal cortex. So this particular area here at the front, And this particular area here, at the medial wall of the prefrontal cortex is called the subgenual cingulate cortex. These are two regions that I will touch base when I discuss some of our work in the clinical population. Then, of course, it's not all structural. So it's not only the anatomical wiring of the brain that is different, but on top of this anatomical wiring, there is pattern of what we call activity or functional connectivity or effective connectivity that emerge. And of course, because the difference in anatomy, the possibility of develop activity on top of this anatomical scaffolding is slightly different. This is just an example of a study that we did a few years back, where we kind of look at states of the brain that are characterized by a specific pattern of activity during the view of an emotionally engaged movie. And this is, you have subjects, so this is each subject pattern of activity, and you can appreciate that there is some synchrony among subjects when they look at the movie. And particularly what we observe is when people engage in emotionally, or there is sadness involved or like that, there is again this kind of frontal, mid-frontal area, and also the lateral part of the prefrontal cortex that is engaged quite consistently across subjects. Just to say again that there is something that is, if we take this evolutionary perspective, that is human specific in how we appraise and we our sensory environment. And then the last thing for this part I want to touch base is that, of course, the way that the structure and the function are coupled in humans is also linked to the manifestation of symptoms in disorders like this, I think, is ADHD. Yeah, ADHD and also schizophrenia. So there is also something in how this function and structure get coupled that seems to play a role in these complex symptoms. Now, so one study we did was to answer this question: what human-specific connection emerge from the brain anatomy? So, is there something functionally specific that emerges on top of this anatomical scaffolding that is different? And we choose the chimpanzee because, as I mentioned, is our closer relative. The issue that we had is, of course, we wanted functional data on the chimpanzee, but they don't exist. So we look around. As you know, it's illegal to do research on chimpanzee around the world right now. So we were able to get from a group in Atlanta some diffusion MRI from chimpanzee and build this connectome in chimpanzee. And then we use computational modeling to try to simulate the activity that will go on top of those connectome and try to study what's the difference that we observe here. So the modeling, for people that do modeling, we did several modeling. This is a reduced one modeling, but we use also the Wilson count that I think is the DCM model that is used at the top of the DCM, at the bottom of the DCM. That is, I guess, this one. And we were assessing a specific property of the dynamics of the brain, and the property is how a change in the global tone of activity in the brain could impact the activity of a single region. No? And we call this the dynamic range, meaning that a region that with a small change in input for the rest of the brain has a big spike in activity, so a bit different in activity. This will have a small dynamic range. On the other hand, a region that have a slower reaction to global change in activity will have a higher dynamic range. And there is some literature linked to the dynamics or the hierarchy of time scale and other aspects suggesting that sensory regions should have a lower dynamic range. So when you have a small change, they tend to react very fast. And associative region or cognitive related region have a slower dynamic range. So higher dynamic range. So what we found is so in human, so each line here is a brain region. So what these two graphs shows is that in human, the way that the brain region change their activity as a function of the change in their input, it's somehow more homogeneous. So that's what we observe in chimpanzee. And as expected, we found that in both species, the blue regions here, mainly the sensory area, have a fast or narrow dynamic range. And on the other hand, the region that are more frontal region or associative region seems to have this more spread dynamic range. So that was as a first observation. And then we looked, okay, what actually is the implication for functional connectivity? There's a common measure of, how regions interact functionally between each other. And we took this network approach where you have the visual network, the somatosensory network, and so on and so forth, the dorsal attention, and these are this yellow and so on. And what we found is actually that because of this difference in dynamic range, again, the chimpanzee having more heterogeneity in the region of dynamic range, the end result is that humans are, more able to connect within and between the networks compared to the chimpanzee. On the other hand, chimpanzee, you can see the peak in functional connectivity happens at different level of general pattern of activity. So for human have a brain that is actually very able to synchronize quite fast compared to the chimpanzee. And we also found that if you take a measure, the graph measure called path length, so you mean to connect from A to B, how many hoops you have to do, and the longer is the path length, the more indirect is the route. So what we found in general is that overall, human and chimpanzee, human have a lower functional path length. So if you are in region E, you want to communicate with region B, you generally need less hoops. So it's a faster communication compared to the chimpanzee. And there is this relationship saying that, a region that have indeed a higher ability to functionally integrate with the rest have a higher dynamic range. It's a bit, I think they're okay, but we were looking across different species. We did the same exercise to see is this really human that have this kind of more narrow or similar dynamics in the activity, and it seems that is the case. So humans indeed seems to have a lower variance in the dynamic range compared to the chimpanzee, the macaque, and the marmoset. Now, then the extra question was, is this connectome, is this different anatomical wiring of the brain really play any role for behavior? And again, we were not able to access data about functional data and behavior in chimpanzee. But what we did, we used this, probably psychologists very familiar with this model, drift diffusion model. And we basically just simply see, okay, to go from once you give an input and it propagates through the brain, what is the time, how much time does it take to reach the threshold of a decision, no? that's a relative simple thing. And what we observe is that so the negative value here, so this is when chimpanzee outperform humans. So meaning the decision is reached faster in the in the chimpanzee brain compared to the human. And when you are in the positive range here, it means that the human scaffolding will allow to, respond or achieve the decision in a faster way. And what we observe is that in the chimpanzee tend to outperform the human in very fast, in tasks that will require fast response. For example, it could be a visual detection or something like this. But as soon there is some integration that need to happen, so the assumption is that will take longer. So when the task will require longer time of integration, the human tend to outperform the chimpanzee. And there seems to be some sort of, once you go down if you want to say the hierarchy of evolution, these relations seem to become stronger and stronger. Okay, that's a little bit the first part of the talk. It's a little bit of background of what makes us human, I guess. And the second part is in what potentially make us sick. And then the second part of the talk is more about, okay, what do we do with this information? Did we study? How do we change? How do we develop therapy with this information? And this is more about the clinical translation that we have done in the lab. So I guess the assumption here is that mental illness can be characterized or can be defined as a functional disorder of functional integration. So that's the premise, also based on what I presented before. And the idea is like, if it is true that we have some sort of problem in integrating information between the regions, or how this global integration affect the region, how can we intervene to normalize this kind of process? So the first case that I want to mention is the case of depression. Maybe you remember in depression, what we observe, this is neuroimaging study and invasive study with electrophysiology, show that the depressed state is characterized by some change in activity, particularly between the prefrontal cortex, a particular region called the dorsolateral prefrontal cortex, and the subgenual cortex. And these, as I mentioned before, that interestingly, two regions that are human specific. So they have expanded, or they have they have evolved much more in humans compared to other species. Another thing before that is interesting to observe is that we all, people in the field often see this kind of image about brain networks, where you have, for example, the vision network in blue, the dorsal attention network in yellow, and so on and so forth at the group level. But when you try to look at subjects, so here is one subject, two subjects, and so on and so forth, there's kind of the overall architecture is still there, the functional architecture, but there is some variability. Now, for example, if you take subject 1 and you focus on the dorsolateral prefrontal cortex, you can see that the way this functional architecture is organized compared to the subject 2 is quite different, no? And that is something that we should also consider when we think the development of therapies. So with these two types of information in mind, what we did with some colleagues here in Melbourne, Andrew Zaleski and Robin Cash, is like, okay, we know that these two regions are misconnected in depressions, and we know that there is some sort of heterogeneity of how the organization of this region happen across subjects. So to try to rebalance this, we need first to find a way to change the activity of one of the two regions, doesn't matter which one, but at least one, to change the coupling between this region. And second, we need to be precise at the individual level. So we develop a method that allow to do that. I will not go into the detail that, but basically what we found is that in people with depression, if the optimal stimulation site using, for example, here we use transcranial magnetic stimulation, will be the site that has the higher anti-correlation between the dorsolateral prefrontal cortex and the subgenual gyrus. So we developed this approach that allow us to identify in each individual what will be the best site in terms of functional connectivity to be stimulated with TMS. Based on that, we kind of move on. And one of the problems that we had was that, well, we didn't have a clinic that was keen to translate and test our approach. So we created one. This is the Queensland Neurostimulation Centre. This is our clinic. So the clinic is a clinic that's been created as a non-for-profit entity to try to really translate cutting edge, or what we think is cutting edge technology into clinical practice. We operate with the approval of the TGA as experimental treatment. So like this is this authorized prescriber scheme. So and the first treatment that we offer was this one in depression. So we offer this personalized treatment of depression. One thing that we wanted also is that it has to be very precise, because you may appreciate there is some difference in this in where you stimulate, and this difference is quite small. So we partner with a company that develop a robot that allow us to give the TMS in a very precise way. And this is Leonard, and this is Leo Marcus, that actually is here in doing a PhD here in psychology, but was a technician. You can see stoked to be the participant for this. This is during the development. But maybe I can give you. He was trying to demonstrate how the robot maintain, the connect, maintain the contact and allow precision stimulation, even when the patient move and so on and so forth. So 3 years on, we have the first results of the clinic that are published. So these is a non-randomized, non-sham, studies, but basically this is real world. So this is the personalized target for the first 61 individuals that underwent the treatment. So the yellow dots are the one that respond. No one really respond 100%, but respond really high. And the more dark dots are people that don't respond. I think the first observation is that it's a little bit like a Christmas tree. There is no particular cluster, so really suggesting that there is some importance of using functional targeting to really achieve some level of personalization. Another thing that we looked is like, did we actually target the right spot? Or is a general effect of the brain? So we look at the connectivity in a subsample, because not everyone had agreed to have a scan after the treatment. This was a research scan, so a lot of people said, I don't want to do it. But anyway, in 28, you can see that we have a specific change in the pattern of connectivity between the pre-treatment and post-treatment. So the treatment is 1 month of TMS. But when you look this is one particular comparison, when you look at the pattern of connectivity between the region and the rest of the brain, doesn't seem to be any change. So it seems that actually specific to that particular connection. And then we looked at, so in the clinical trials that you see in depression, people generally meet very strange inclusion criteria. In real world, things are a little bit different. And so we came up with, we didn't come up, we adopted at the beginning of, at the start of including people, we adopt a kind of subdivision of patient in three groups. So group one is people that will meet the criteria for a randomized controlled trial. So group two are people that have major depressive disorder, but they also have a comorbidity, like could be anxiety, could be obsessive compulsive disorder, and so on and so forth. And group three is a group of people that have a major depressive disorder, but they also have a neurological condition, Parkinson and Alzheimer, and so on and so forth. So, and what we observe is that, well, everyone needs to go. So MADRS is a score, is a scale for depression, and HAM-A is a scale for anxiety. And what we were able to observe is that overall, everyone seems to do, the symptoms seem to improve, but there is a much greater improve in people that have actually major depressive disorder only, compared to people that have, some comorbidity or co-occurring neurological disorder. And this is also true when you vary from age, because people that have a neurological disorder seems they are generally older than people that in group one. What was we were kind of reassured is that the results for group one are really striking. I mean, response is defined as at least 50% reduction in symptoms compared to baseline. And remission is basically when people don't meet any more clinical criteria for depression. And what we observe is that 53% of the people, these are people with treatment-resistant disorder, they didn't respond to medication, didn't respond to psychotherapy. 53% of them show a response, and overall 74% show a response, in which 53 had remission. This is really a striking result. Generally, in clinical trial, what is reported is between 45 and 50%, around 50%. So we have an increase of about 20% compared to what is the baseline of the clinical trial. Okay, that's for depression. And then I'm going to present you a similar story for obsessive-compulsive disorder. So obsessive-compulsive disorder, many of you know, is a recurring, very severe condition characterized by obsession and compulsion. Interestingly, again, going back to my evolutionary blah blah at the beginning, was that we have this region in the frontal area, this frontal pole area, that is has been particularly associated with obsessive compulsive disorder or obsession and compulsion, and particularly the connection between this region and the striatum via the thalamus. There is several lines of evidence. So there is neurosurgical evidence. This is a study from someone in our lab, DBS study showing that the stimulation of a white matter tract connecting this subcortical structure with this anterior prefrontal region improves symptoms of obsessive compulsive disorder. This is another study, a very pioneering study from a Dutch group, showing again a strong association between deep brain stimulation in the striatum and change in the frontal activity. And this is a tractography study from the group of Andreas Horn at Harvard, indicating what are the fibers that are linked to improvement in OCD symptoms following DBS. And again, these are fiber in red here connecting, this striatal region, the thalamus, and this frontal area of the brain. We also did some neuroimaging, replicating this early study from a person here in Melbourne, Ben Harrison, showing that there is a hyper connectivity between the nucleus accumbens, so a structure of the striatum, and this anterior frontal region. And then there is also a hypoconnectivity with the lateral, more cognitive system between the putamen and the lateral prefrontal cortex. This is the results of their study. This is the results of our study. There is some thresholding about statistical threshold, but they are pretty much the same, similar region, very similar region, the same system when you look at the system level. So you have this anterior part of the orbitofrontal cortex and this lateral part of the prefrontal cortex implicated. We didn't find any change in structural connectivity, so it seems that OCD, it's probably more like a functional disorder. There is not strong different. We are not only this is not only us. Other people suggest that there is not huge structural change in the OCD in this pathway. And we were trying to use, again, the rationale was try to use TMS on these prefrontal regions. We not go into detail, but there was also a reason about the dynamics, how you change the dynamic using a specific TMS protocol. But the idea was you decouple the activity of this frontal area with the subcortical region of the striatum. That was the rationale. So you reduce this hyperconnectivity or hypercommunication between. The study was published, so just to guide you through, it was a clinical trial. This was a randomized controlled clinical trial. So we have two parallel groups, sham and active, with, again, 20 sessions of inhibitory stimulation of this area. This is our target compared to other target in the literature. So people, so we have 56 people with clinical OCD that got randomized, 24 in 24 in one group and 26 in the other group. They were reasonably balanced. They were quite young, so around 30-year-old. For people who are familiar with the Y-box score, that is clinical score to assess obsession and compulsion, is a quite moderate to severe symptom of OCD, a score of 25. And with preserved IQ. Again, OCD, this is not new, but OCD is not associated with a cognitive impairment. So we stimulate, this was a neuronavigated stimulation. Here is the spot for, I think the blue is one group, the orange is the other group. This is the behavioral results. So we have sham and active, a baseline, post stimulation, so means a month after, and then a 6 months. So unfortunately, what we saw is that there is no difference between the sham and the active. Actually, if anything, the sham did slightly better. And there was no difference in the connectivity between the target area of each individual and the nucleus accumbens. So you can see this is baseline and post in the sham and the active, so no difference there. We also found no difference in the area that we stimulated. And I think this was probably associated to one explanation. It's always difficult to really, but we use a very low intensity because it's very painful to have brain stimulation in this area. So we use 70%. That is a very low intensity, so it probably does explain. But everyone seems to do better regardless of the stimulation. So if you go back here, everyone seems to reduce their symptoms regardless of active or sham. So we look at the main effect, of getting better. And what we observe is that when you get better, your integration between the default mode area and the frontoparietal system. So default mode, for people that are not familiar, this network involving interoception, rumination, and this kind of thing. So the connection between these networks and the frontoparietal system, that is more the goal-directed network, is improved. And the internal connection within the default mode is decreased. So it seems that this probably could be a substrate of why people get just better in symptoms. And then my last slide is about a little bit presenting to you what do we do in Queensland. So I think the lab does a little bit of, we have basic research. So we have a lot of network in neuroscience, understanding how the brain region communicate and so on and so forth. We use imaging, we use behavior, and we use computational modeling. We do some clinical research. Again, this is mainly in the form of taking patient and see what's the difference between patient and control. We have DBS trials where we are now also able to measure the activity in the regions where we stimulate and also put people in the MRI, so we can actually assess online how to stimulate a region affect the brain. We use a range of neuromodulatory technique. That this is our new focus ultrasound laboratory. But we also have TMS and that we use quite a bit. And also one thing that I'm quite excited is we are trying to, before running into trial, we are trying to develop a platform that is a computational modeling platform that allow us to modulate various parameters, can be the strength of excitation, the strength of inhibition, and so on and so forth, to try to decide what what treatment, in addition to the data, could actually result in the best outcome. And I think this is something that I'm quite excited. And then, of course, we finally have a clinical platform that will allow us to collect data for this stream, but also, translate what we found here into, into the clinic without too many delays and so on and so forth. So I didn't do all this alone. This is the lab, the core people in the lab. The work that I present you with the chimp and so on and so forth was led by James Peng, that is here now in Monash, and then modeling work with James Roberts and Michael Breakspear, and again, especially the depression work, clinical work with Andrew Zaleski and Ronnie Cash. Thank you very much.
Wow, that was amazing. I was so blown away from how you go from basic fundamental neuroscience into doing something that is so impactful in the clinic. So that was a beautiful demonstration of the journey from fundamental work to impact in neuroscience. Amazing. So I'm going to have the students to ask the first questions. Okay?
Yes, thanks for the amazing talk. I want to ask a question from your first part, where you compare the human from like human and chimpanzee. What kind of tasks did you use for comparison?
So we didn't use tasks because we couldn't access tasks. So basically, we use this proxy. We use this brief diffusion model to simulate the task. Like what will be the threshold of reaching a decision, assuming this connectome, this pattern of. So we didn't actually have access to the task. There is Martin van der Hoevel now has obtained some task data on chimp that also human have done. This is very old data, so we are going back in the 60s or 70s because and we are starting to look at all of these tasks, but it's very sketchy data about it. Yeah.
So I was just wondering, because if the human and chimpanzee have a different learning ability, so if you're comparing, so how can we compare, how can we decide the learning rate? Maybe if you are giving simple tasks to chimpanzee, but difficult tasks to human, it's literally hard to compare the activity.
Yeah, this is really not learning rate. This is just input output that we had in this particular study. But you're right, in terms of learning the specific task, let's say, well, we know that, for example, chimps take longer, even a monkey, when people that work with monkey in the lab when they have just to learn 1,000 eye movement and so on. It's a long process. So they get faster in stimulus response, but once you need integration and probably learning involved that type of integration, then you want to outperform.
I'll just follow up on that about the speed of response, because I was thinking, you had the humans are better after 500 milliseconds, and that's kind of the average of almost any response that you do in any behavioral task. So what is earlier than that? Are these motor responses or like with finger presses, or are they mostly eye movements for the monkey?
So in chimp, where this was this person, James Rilling, that is in Atlanta, what he told us generally what they did is mainly motor response, not like do reaction time.
Reaction time.
Yeah, very, very basic. And apparently we look, but there was early science paper, again, you go back in the 70s, and that showing that actually chimp outperform human in very simple tasks. But then as soon there is learning or more complex thing, then things break. And maybe it's this integration difficulty. Yeah.
More student questions? Or we go to Jay? Oh, yeah.
Recently, I discovered that axolotls can regrow parts of their brain. I don't know whether we can kind of harness this new technology in this lab, or it's, I know it's kind of different, but I was wondering about your thoughts on this.
Well, I didn't know that. I will look into it. It looks interesting that they can regrow part of the brain.
Yeah.
Oh. Yeah, sorry, I cannot offer.
That's it. Yeah. It's there were some experiments being done on axolotls and seeing if we could kind of see why they can do that, and seeing if it can help humans who have had injuries.
I'll try to understand what's the mechanism of regeneration. Yeah.
How big are those brains?
Yeah.
That's well, thank you.
Yeah.
How big are their brains?
Axolotls are tiny, but they can regrow limbs, they can regrow parts of their brain, and it's fascinating because they're vertebrates. Yeah.
Cool. We're going to
Yes.
student questions. Okay, you can ask later too. Jay.
Can I ask, with your depression data, what was the period, the post assessment? How long post TMS was that?
Within 2 weeks after the end of the TMS.
Right. And given that you didn't have a sham condition, how confident are you that you're actually going to find something?
Yes, that's a very good question. So we are running a clinical trial now. Finally, the NHMRC, after 4 years of trying, gave Robin actually the money for the clinical trial. And look, I was always a little bit skeptical about things, but when you look at this one. When you look at the reduction, especially in group 1, where we have strong benchmark of randomized controlled clinical trial, non-personalized, and again, this is around 45-50% response. I think maybe there is something. I will not say, but achieving 74 is like a almost 25% improvement. There is another thing that is very fascinating work of Luke Cheng, is someone at Dortmund, about placebo effect. And he showed that, for example, the level of perceived technological thingy in your boost the placebo effect. So huge. Like we have the MRI, we have the fancy robot, blah, blah, blah. Does it improve 20%, 25%? Maybe. I actually, we don't have an answer for that. I mean, it could be. Yeah, you have to run a trial. Andrew.
Yeah, thanks, Luke. That was great. I was wondering with your comparisons between the different primates.
Yeah.
Can you tell us more about humans versus the chimps, the macaques, and marmosets? Were they raised in a zoo or a lab? And thinking about, I don't know the extent to which those structural connectivity measures would be changed by early life environment or ongoing life environment. And that could be part of the effect beyond the species differences, the way they were raised.
Yeah, I think that you raise a very good question. So we don't have a lot of details about the chimp. They were raised, from what we know, we were raised in this kind of zoo where they can go around. They call it nursing something, but they can go around like that. One thing that I would say, because the quality of the image, we use a technique called deterministic tractography, that is quite stringent. So the type, there's probably more type 2 error in the detection, the connection, the type 1 error, meaning that you probably detect just the big fiber, the one that are, don't quote me on that, but I would not expect that they're highly modulated by the environment, like a corpus callosum, the anterior pharynx, all like that. So it's possible, but I don't know how much. I don't have an answer for that specifically, but I don't, I don't know about the environment. One other thing that I would like to say is that I don't want to give the impression that, I give, animal models are useless in this. I think animal models can really help in psychiatry to unpack, fundamental mechanisms of synaptic deregulation or like that. They can unpack specific pattern or understanding neurobiology, but maybe when it comes to, really understand what type of dynamics or high level things are involved in some symptoms, there is some limitation that need to be. That's what's the reason. Yeah.
I think it'd be interesting with Nick Martin and others, identical twins.
Yeah.
You've got that using the same structural connectivity.
Yes.
And I know that a published study, how similar and different identical twins are, in terms of genes and environment, to give you some sense about variable environment between pairs, because there are some areas that are invariable and there's some that are highly variable between identical twins.
Yeah, we did one thing with Andrew and Robbie. We took the human connectome, this is another paper, but we took the human connectome data, and if you have 1 year pre and post scanning, and then we looked at on unrelated people, dizygotic and monozygotic twins, about the stability of the site of stimulation. And what we found that actually is there is a progressive divergence. So in a year it's quite stable, but it's more similar the site in monozygotic twins. Then it move, there is more heterogeneity in dizygotic twins, and there is much more heterogeneity across in the normal population. So it seems that there is more heterogeneity at baseline, and also the stability, it seems more stable in monozygotics than the general population. So there is definitely some genetics involved in environmental effect in that.
Just a clarifying question on a similar topic on the targeting of the stimulation. You mentioned in passing, I think, that it was targeted based on anti-correlation between two brain regions.
Yes.
Did I hear that correctly?
Yes, that is correct. Yeah.
What was the rationale for kind of using that anti-correlation as your sort of metric?
Yes, that's a good question. So the anti-correlation came from an early study from, I always pronounce her correctly, Myber in DBS and PET. And then later on, Michael Fox and others showing that the level of anti-correlation between the subgenual and dorsolateral prefrontal cortex is proportional to the symptoms. That was the rationale. And I am I'm less convinced that the anti-correlation per se is the connectivity, yes, but the anti-correlation, I'm not 100% sure. So meaning, I think it's more on the absolute value that we should look at. In fact, if you maybe you pick it up on that, I clever try to hide that, but you can see that between pre and post. So this is the level of anti-correlation, no, between these two regions. And you can see that actually in post, where people tend to do better, the anti-correlation decrease between, between these two areas. So I think there is something with the magnitude of association between the signal in these two regions. I'm less and less not super sure about the anti-correlation. The anti-correlation story is sexy because, as I told you, they form all the interoception and like that, and the other is front right, and the story is like, well, there is intrusion of the symptoms into the cognitive machinery, and the more you dissociate that, you get better. Yeah, I'm not 100% sure. But then if you look at the OCD results that the last slide of the OCD results. Here actually the story seems to hold, where when people get better, they increase the pattern of communication between the default mode and like that, and they reduce the intrinsic connectivity. So interesting.
Okay, I think we might
Yeah.
have to wrap up.
Yeah.